Well, welcome everybody. I'm Bill Durgan. I'm the provost here at SUNY IT, and this is the second presidential lecture this year. We're delighted to have with us Professor Terri Camesano from Worcester Polytechnic Institute. Terri is a professor of chemical engineering and also has an affiliation with the civil engineering and environmental group down there as well. Terri's going to talk to us today about bacterial adhesion, which is her principal research area. And I thought this was an especially appropriate talk in light of the things that are going on here on this campus in nanotechnology, because Terri's work touches not only on the health sciences, but also on nanotechnology and engineering. So it's quite a broad-based disciplinary focus here. One thing that I will tell you is that Terri is the principal investigator on an NSF IGERT award, and that may not mean too much to any of you, but an IGERT is a major NSF program. It's focused on, in this case, on biofabrication, making things, using living cells to do that. IGERTs are tough to get. I know that WPI has been working on this for a number of years to get an award like this, but it's $3 million over 5 years to support graduate education in the interactive graduate research area. And so it's a very, very prestigious award that Terri's the person in charge of that entire program. So a lot of success there. But in any case, let me ask Terri to come up here, and we'll listen to her presentation, and then we'll entertain some questions. And I want to thank you all for coming. This is a great turnout. So Terri, this is your presentation.
Good afternoon, everyone. Thank you very much for coming today. I apologize that I have a really bad cold, so I hope that you can hear me. If you can't hear me, just raise your hand in the back, and I'll try to get a little closer to the mic. It's really a pleasure to be here today. Your provost, Bill Durgan, was an administrator at WPI when I joined there almost 13 years ago, and was really instrumental in helping get my research program started at the beginning of my career. So it's a pleasure to be able to speak to you today about my research. It's also a pleasure to be back home. I graduated from Whitesboro High School, and I'm from this area, so it's really nice to be able to come back here and tell you about some of the work that we're doing at Worcester Polytechnic Institute. Today I'm going to talk to you about two of my favorite subjects, which are cranberries and bacteria. And if you've never thought of those two things as going together, then maybe after today's presentation you'll understand a little bit more about why I'm grouping them together. So this is a really good time of year to talk about cranberries, because October is the cranberry harvest season. If you've never seen these in person, I think you've probably all seen the Ocean Spray commercial with the two guys in the cranberry bog wearing the boots and everything. And if you haven't seen it in person, I highly recommend that you go, because it's very interesting, very educational. 90% of the world's cranberries come from the United States, and in particular coming from Massachusetts, it's really important for us because Massachusetts is the third largest state producing cranberries in the US, so it's really important for our local economy. Cranberry production has also been really increasing, so you can see from the 90s to now, US cranberry production has been increasing, and in part it's because there's been so much marketing being done related to the health benefits of cranberries that people are just becoming really interested in all kinds of cranberry products. So one of the reasons why cranberries are so beneficial for a number of different health reasons is because they contain these chemicals called polyphenols. Polyphenols are found in a lot of different fruits and beverages, so besides cranberries, also things like apples, pears, cherries, even chocolate, red wine, of course. But what's really interesting, and you'll learn as I go further in the presentation, is that cranberries are pretty unique in the exact type of chemicals that they contain, and that's why some of their health benefits are unique even compared to other things that we think of as being healthy, like apples or red wine. There are a number of different health benefits of cranberries that people are actively researching in today. My work relates to anti-adhesion, and I'll explain what this is more clearly in the next few slides, but things related to adhesion of bacteria to different surfaces in your body. So urinary tract infections are caused by pathogenic bacteria attaching to either your kidneys or cells in your bladder. Some ulcers are caused by bacteria attaching to the epithelium that lines your stomach. And gum disease can be caused by bacteria attaching to gum tissue. So this is one class of diseases or conditions that can be affected by cranberry. The other ones I put on here, I'm not an active researcher in, but I thought you just might be curious about. So I have colleagues who are working on anti-aging. I'm sure you hear a lot about free radicals and fruits and vegetables that can protect you from that, and cranberries are loaded with them. Cranberries are known to have heart benefits because they have so strong antioxidant properties. And one of my good friends, a collaborator at UMass Dartmouth, has been studying the anti-tumor activity of compounds in cranberries. So this is just kind of an overview, and I'm going to be talking a lot more specifically about my work, which is in bacterial adhesion. So I'll talk to you a little bit about why people think you can use cranberries to prevent urinary tract infection, and talk to you about how we're thinking to detect these compounds in urine, how to identify the active ingredients. So urinary tract infections are a very, very common health problem, with over 11 million patients being diagnosed with a bladder infection, which is cystitis, in the United States every year. So kidney infections represents a smaller class of patients, but they can be more difficult to treat, they can be more complicated, and they can lead to more long-term health effects. So both of these together are extremely expensive for both men and for women, resulting in many, many doctor visits and in the prescription of antibiotics. This becomes an issue in itself because I'm sure you've heard about the problem of antibiotic resistance. So this was actually one of the reasons why I wanted to get into the cranberry field, was to work on something that was a little bit of a side path, if you will, rather than a traditional antibiotic. And in fact, urinary tract infections are particularly a problem for some women. So there are some women that are constantly either getting a urinary tract infection, being treated for one, just get over it, and then get another one. So this recurrent infection seems to be another group of type of bacteria that infect these women, and traditional antibiotics are failing as a treatment for that. So I mentioned that I'm really interested in bacteria. I've been studying bacteria for a very long time. I don't know what you picture in your mind when you think of bacteria, but if you look at this diagram, this represents one E. coli cell. And these hair-like projections that you see all around the cell, they're actually proteins. And because of these proteins, they are the way that bacteria can latch onto either cells in your body, or a catheter if you had to have a urinary catheter, or gum tissue. So the reason that we get sick from bacteria is because these proteins on the bacteria are able to attach to cells in our body or biomaterials that are in our body. I've been really interested in this one particular type of E. coli. One of the proteins has been named P. fibria, and that's been identified to be responsible for 90% of the cases of pyelonephritis, or an acute kidney disease. So this simple cartoon is just going to illustrate to you a little bit about how the infection occurs. So imagine that these are epithelial cells in your body. Bacteria come along with their hair-like protein projections. and they can attach onto the uroepithelial cells. So one of our big interests is preventing this adhesion process. We can't really prevent the bacteria from being there. Bacteria are normally present, they're present in our bodies all the time, but we can prevent the pathogenic bacteria from attaching to uroepithelial cells. So I want to just take you back a little bit to some of the early studies in this area, building up to, as Provost Durgan mentioned, I take a nanotechnology approach, but I think before you jump into how nanotechnology can help in this problem, it's interesting to learn what was done before then in the field. So this is not my work. This is some work from the early 90s, electron microscopy on images of different types of bacteria. This one you can see has no hairs on it, so this would not be a pathogen. If this bacteria was coming in contact with your cells, it wouldn't be able to attach, it would just be discarded. And this one with the hair-like projections would be able to attach to epithelial cells and would be able to cause a kidney infection. So one of the first experiments that my former student did, she's now a postdoc, she did this work when she was an undergraduate, was she incubated epithelial cells with the bacteria. So these large circles that you see, that's the cells that line your bladder. That's epithelial cells in your body. These small, darker cells that are more rod-like, those represent the bacteria. So this was with the pathogenic bacteria. In this case, there was no cranberry juice present. In this case, there was 10% cranberry juice present. And in this case, there was 27% cranberry juice present. So what you can hopefully see, I know it's a little bright, is that when there was no cranberry juice, there are tons of bacteria that attach everywhere on the epithelial cells. When there was a little bit of juice, there was a few bacteria on the outside, but definitely not as many. And in the 27% juice, very, very few bacteria. It's interesting, the reason we picked 27% juice is because the cranberry juice that you buy at the store, if you buy something called cranberry juice cocktail, it is in fact 27% cranberry juice. Cranberries by themselves are very bitter, and so you can find 100% juice, but it's not very palatable. Most people don't like to drink 100% juice, so usually whatever you buy in the store is actually 27%. We decided to make that the upper limit in our tests because we thought whatever we learn from our lab studies, we want to be able to apply this to real people going to the grocery store and buying juice. And so if that's the maximum dose that people would be able to get and be able to drink, then that's what we kept as our maximum. So this is nice. I mean, for my student, this was a nice result. It's very visual. It shows you really well what's happening, but we have an engineering background, and so for us, it's nice to look at a picture, but we really want to understand why. Like, what is the mechanism here? What is cranberry doing, either to the bacteria or to the epithelial cells or to that overall interaction to make this process occur? So how are we going to get mechanistic information? I think a lot of you in the room are so young that you have no idea what this is. So let me tell you, this is a record player that we used to use to listen to music a very long time ago. And a record player worked by having a very sharp needle that moved along the grooves in your record, and that's how it played music. This I'm sure you will recognize, which is a tennis ball. So a record player and a tennis ball are a very simple way for me to represent the atomic force microscope, which is a tool that I use in my research. In the simplest way to think about it, an atomic force microscope is a record player needle, something very sharp, being scanned over a sample surface. This happens to be some biological surface, a cell with different receptors. But what you can do with this microscope is you can trace out the topography of your sample, so you can get a map of what your sample looks like. And you can also measure forces, intermolecular forces, between this needle and whatever your sample happens to be. So the slightly more sophisticated picture looks like this. We have a very sharp probe interacting with our sample, and using a laser that shined on the probe and that light being captured in a photodiode detector, we can tell everything about our sample. We can tell what our sample looks like, and we can measure the forces. So if I'm interested in studying adhesion between the bacteria and the epithelial cells, this would be the way for me to do it, because I could directly measure those nanoscale adhesion forces. And this cartoon represents essentially what we tried to do in our experiment. We took epithelial cells that line the kidney or the bladder, we've done it with both, and we took one bacterium, so one E. coli bacterium, we put it on that tip, that record player tip, and we measure the forces of adhesion between the two. This is an electron microscope image. If you can see right here, this is a single bacterium of E. coli. So we bring this single bacterium into contact with cells from the urinary tract, and we measure the forces of adhesion. This represents really a few years of work. So I had an extremely motivated and extremely patient graduate student who worked very, very hard to be able to get individual bacteria attached to that AFM tip so that we could finally get at these measurements. Oops. So let me show you what our results showed at this point. This plot shows the force of adhesion as a function of the concentration of cranberry juice in the solution. And the black dots here represent the pathogenic bacteria. So this was the E. coli with P. fimbria. When there was no cranberry juice, the force of adhesion was very, very strong. So even though this is in the nanonewton range, this represents a very strong adhesion force. And as you add more juice, 2.5%, 5%, and up to 27%, those adhesion forces essentially disappear. So we were able to demonstrate for the first time that the nanoscale adhesion forces between E. coli and your epithelial cells were directly impacted by cranberry juice cocktail. The one that doesn't look too interesting down here at the bottom, these open symbols, those are the non-pathogenic. So those are the E. coli with no hairs. Cranberry juice doesn't do anything to them, but that's because they're already not very adhesive. They don't have the right molecules on their surfaces to be able to participate in adhesion or cause an infection. So the next thing that my student wanted to do, we got these results, and this was really, really exciting for us because we were the first group to directly demonstrate that these adhesion forces were affected by cranberry juice. But this was all done in a lab with juice from Price Chopper right out of the bottle. And the next thing we said was, what happens if a real person drinks that cranberry juice and the cranberry juice passes through their body? Is there something still left over after cranberry juice is metabolized or partially metabolized by the body? And what can that do in relation to adhesion? So that was the next question that we tried to answer. And so we moved to what I would call is a very preliminary clinical study. So remember, I'm a chemical engineer, I'm not working in a medical school, but I still wanted to get some clinical information. So what we did here is we had a volunteer. This was actually done in collaboration with Rutgers University. So the volunteer and the sample collection was done at Rutgers. The volunteer would drink cranberry juice cocktail or water and then collect their urine every 2 hours. Then the urine samples were sent to WPI, and we used atomic force microscopy to try and understand whether we could detect molecules in that processed urine that were anti-adhesive. We had to simplify the study design a little bit for this, so we took out the epithelial cells, and we decided for this part of the study we would just probe the E. coli themselves and their proteins on their surfaces. So I alluded earlier to this problem of antibiotic resistance, and I'm sure that you all hear about this. Maybe you even think about it in your daily lives, because the incidence of antibiotic-resistant infections is just growing. So from 1980 to the present, you can see MRSA. I'm sure you hear about MRSA. This is methicillin-resistant Staph aureus. There are other types of microbes that are resistant to other commonly available antibiotics. The problem that we have is that bacteria can evolve in 30 to 45 minutes. So their lifetime and their ability to evolve is much, much faster than any other kind of organism. And so if one bacterium has a plasmid that protects them from being attacked by an antibiotic, they will transfer that to other bacteria, and quickly the whole population becomes resistant, and that's why we don't have anything that can treat these infections. Unfortunately, the development of antibacterial agents is really lagging, and so if you look at antibiotic development in the United States compared to the 1980s, it's really, really low. There's a number of reasons for this. I think one of the reasons is that antibiotics are not as lucrative a field to go into compared to certain kind of drugs that people would need to take every single day, and other scientific reasons as well. But for whatever reason, we don't have as many choices for antibiotics, and yet we have more and more need for either antibiotics or alternative kinds of therapies to prevent infections. Cranberries, I think, are really, really interesting here, because the way that cranberry works against bacteria, it actually has nothing to do with antibiotic resistance. Cranberries don't kill bacteria. I wouldn't use it to clean my countertop or to sterilize a cutting board. It doesn't kill the bacteria. But because it doesn't kill it, it works in this purely physical way of preventing the attachment. That means the bacteria are not going to be able to easily develop any kind of resistance to it. In fact, I don't know of any study that showed that bacteria could prevent a resistance to attachment, because it's really a physical process. It's not a biological process the way that antibiotics work. So this scary table just shows you some of the clinical strains of E. coli that we work with. All of these E. coli strains came from patients with infections. So some of them came from people with bladder infections, and some of them came from people with kidney infections. And they were already known to be antibiotic resistant at the time when the surgeon removed these from someone's body. So what we wanted to do was to show if cranberry could have some activity against these antibiotic-resistant bacteria. So this is the result of what I called that preliminary clinical study. What you're seeing here, in every case, is adhesion force, and the blue bars represent adhesion in the urine when the volunteer drank water, so blue is water. And pink represents adhesion in the urine when the volunteer drank cranberry juice. And I have this, I have a plot like this for every strain that we looked at of E. coli. Actually, all the strains had very, very similar behavior, which I think is encouraging. In all of the cases, for any of the E. coli, you can see the pink, the cranberry bars are always lower than the water bars. So with our atomic force microscope, we could measure adhesion forces in the urine, and we could show there was always a decrease in the urine of the person drinking cranberry juice compared to when they drank water. And these two are antibiotic-resistant bacteria. This is another antibiotic-resistant bacterium, and then we looked at two other strains. I'm going through these quickly because the results are generally all the same. In all cases, the adhesion in the urine was lower when that person drank cranberry juice cocktail. So this was encouraging to us, it was interesting to us, and it made us want to go a little bit further with our clinical trials. So if we were in a medical school, they would have looked at that preliminary study I did, and there would have been quite a few criticisms, valid criticisms. One is that we only had one subject, and the other is that our control, which was giving the person water to drink, maybe is not the best control. I think you probably heard of the placebo effect. So if you think you're taking a medicine, maybe you get a little bit better even if you didn't take a medicine. So we had enough preliminary data from that early study that we could apply for more money, and we were lucky enough to get more money to do a better clinical study. This time we decided to do a study on our own campus. So we did this at Worcester Polytechnic Institute. We decided to do this just with women, since young women have a lot of problems with urinary tract infections, and we knew it would be easy to enroll volunteers for our study. So we ended up with 20 volunteers who completed all of the trials. And here we partnered with Ocean Spray. You probably know Ocean Spray is a producer of cranberry juice and products. They didn't give us money for our study, but what they gave us was a beverage that they produced in-house that was a placebo. So it looked exactly like cranberry juice, and as far as we know, it tasted just like cranberry juice. So when the volunteers were drinking it, they didn't know if they were drinking the cranberry juice or if they were drinking the placebo. So this is just a better way to design your clinical study. And we asked these volunteers to drink a certain amount of the beverage and then to give urine samples at different times. We also tried to expand the time points here because we're still really interested in knowing, like, how long does this effect last in the body? That's still a question. So this is the results when we combined all of the 20 volunteers together. So when we combined all the volunteers together and we looked at, these are two different E. coli strains. This is a gram-positive bacteria, Staphylococcus aureus, which is another pathogen. And we measured the adhesion force. What we found is that 2 hours after the person drank the cranberry juice, so if you just look at the first bar to the second bar in each case, 2 hours after consuming the juice, there was usually a really good benefit or a decrease in adhesion. What we found that we were a little bit disappointed about is that we couldn't see that effect 8 hours later in all of the cases. And what we also saw here, compared to the last set of data I showed you, is that there's a lot more variability in how the different bacterial strains behaved. So when we only looked at that one volunteer and 5 different strains of E. coli, it looked like they all were having a similar effect. When we went to a more complicated study design, you can see these 2 E. coli behave different from one another, and the staph bacteria, which is a different kind of bacteria, also behaves really different. So I would say it's promising. It's promising that 2 hours after beverage consumption, you can see this reduction in adhesion. But it's a concern whenever you're thinking about developing this into a therapy that 8 hours after, we couldn't detect much of a benefit. So what we tried to do here, what we tried to do at this next stage was say, okay, we've already done some clinical studies, we have enough of an idea of what's going on. Maybe we need to go way back, really go back to the lab scale, more on the mechanism side, and think about what are the compounds in cranberries that are responsible for these benefits. Because cranberry juice contains so many different kinds of compounds, and when you give it to a person to consume, and you're not able to control everything in their diet, it just becomes very challenging to try and interpret the results. So here we decided, let's go back to the lab scale a little bit and look at the active ingredients in cranberry juice. This doesn't come from my work. This was known maybe 10 or more years ago, that the compounds in cranberry, of the 150 or so compounds, the ones that were believed to be beneficial against adhesion properties were these compounds called PACs, or proanthocyanidins, but I'm just going to call it PACs. And there's 2 different types chemically. And cranberries are believed to be unique. So the USDA has tested this on all different fruits and berries, and what they found is that cranberries are unique compared to any other fruit that's out there. The only other one that comes close is blueberries, but blueberries are believed to have them at a much, much lower dose. So everyone in the community has kind of latched onto this, because if this is the unique molecule, maybe this is the reason why only cranberries are being promoted for urinary tract health and all these other health benefits. Of course, it's always more complicated. Well, as you'll see, the issue is definitely more complicated. So for a while, people were only studying this one molecule, and now the tables are turning a little bit, as I'll show you. What we decided to do here was to get at that question of how long does this effect last? So when I showed you my clinical data, I said everything looked great at 2 hours. It did not look so great anymore after 8 hours. And so what my student decided to do was to actually grow the bacteria in a solution with food, but also with a little bit of cranberry juice, and to extend that out for long periods of time to try to see, did being in the presence of cranberry juice impart some long-term, maybe reversible, maybe irreversible changes in the bacteria. So we would culture the bacteria in growth media with juice, and then we would take a little bit out, put it into new media, take a little bit out, put it into new media, and keep this going for as long as we could, basically. And what we decided to do, because we wanted to have a lot of data quickly, is we put the atomic force microscopy on hold for this part of the study, and we said, let's just look at a really simple assay. Do the bacteria form a biofilm? So if they're very adhesive, they'll form a collection of microbes together, which is called a biofilm. And you can see that if you add a dye to the solution, you can actually see it. So what you see here, there's nothing yet. They haven't had time to grow. They finally start growing 24 hours, 30 hours, and then the reason it becomes lighter is because eventually they don't have enough food and they start to die. So this is a simple assay. But what she was able to show is that the longer you kept the bacteria growing in the presence of cranberry juice, the less they were able to form a biofilm. So absorbance is the way that we measure, if I go back to this picture for a second, if I want to have a way to measure how purple this is or how dense this solution is, I just use a spectrophotometer and I can quantify that very easily. So when the absorbance is high, that means there is biofilm present. When it's very low and flat like this, that means there's no biofilm present. We did this for cranberry juice cocktail, and we did it just for that PAX compound. So here's where we had Ocean Spray help us out again. They were interested in seeing how do their PAX behave compared to the whole juice. So we got some isolated PAX from cranberry juice. And we did these biofilm studies for bacteria grown 1, 4, 8, and 12 times. And then after 12 times, we put them back into media that had no juice, and that's what we're calling the reverse. So we found two really interesting things here. One is that the effect was completely reversible. So even the bacteria that had been grown up for 12 culture periods in cranberry juice, they couldn't form biofilms. When you took them back into regular growth media with no juice, they could form biofilms again. So whatever the cranberry juice is doing, it seems to be more like a physical thing. It's a transient thing. It's not permanently changing their ability to adhere. That has some implications for how you would use this as a therapy or a treatment. The other thing we found that has gotten us into a little bit of trouble now is that the PAX, so what you notice is that none of these lines came down as low as the juice, and none of them sort of flattened out at 0. The PAX by themselves prevented the biofilm formation a little bit, but not as well as the juice. And so when we have presented this and when we published this, some people read our publication, or they read maybe just the abstract of our publication, and then they write an article saying cranberry supplements are not as good as juice, or they will say, you know, WPI professor says that supplements are not effective. And of course, that's not what we've studied, and that's not what we've said, but you can kind of understand now how people may misinterpret your results. So what we did find was that the packs by themselves were not as effective as the whole juice. We don't completely understand that, and for us as scientists, it's really interesting. Like, we want to know why. Why is the juice more effective? Is it because there's other compounds in the juice besides this packs that have been focused on for so long that maybe they have other beneficial properties? Or is it even something about the juice environment? So all of these chemicals can break down easily in the presence of light, or due to temperature, or how long they've been stored. So is there some way that the juice is more protective, or is it that there's other compounds in the juice that are beneficial? It was a little bit scary, but at least it gave us a lot more research questions to pursue in the next phase. So what we've been doing very recently with our most recent grant is we're working with a chemist. So I mentioned I'm a chemical engineer. I do a lot of the physical and biological stuff, but I'm not the one who's going to go and isolate those molecules and tell you exactly what those molecules are. For that, we needed to work with a natural products chemist. And so my friend Kathy Netto at UMass Dartmouth, she starts with the berries, so it's even more simplified than the juice. She starts with the berries. She isolates out all of the different fractions, packs and non-packs and a lot of other combinations, and she gives the samples to us, and then when we do our studies, hopefully we'll be able to be more conclusive about the role of different molecules in the juice. And you know, what I'm hopeful about is that we're going to identify new types of molecules in the cranberry that have health benefits. So just to kind of summarize, I know I had a lot of information in here. This is something that we've been working on for at least 8 or 9 years. The most important clinical questions that I think people need to follow up on are, if you were somebody who wanted to take cranberry juice as a therapy to prevent infections, it's not very clear right now what should be the dose, how much juice should you drink, how often should you drink it, and then of course, what is the role of the non-packs, so the chemical molecules that have not been as well studied, what is their role, and of course, what other diseases or conditions could be treated by cranberry. I think, you know, if we think about infections very broadly, there's probably a number of other types of infections where cranberries could be of some benefit. And as an important science question, we'll be partnering with chemists to really understand what are these other molecules in the cranberry, and do they have anti-adhesion and antibiofilm properties. So with that, thank you very much for listening today. I'd like to acknowledge some of our collaborators. I mentioned I've worked extensively with Dr. Amy Howell. She's at Rutgers University in the Blueberry and Cranberry Research Center. I also had a visiting professor from Spain who spent some time in my lab, and she was instrumental in getting this project going. I still work with Professor Netto at UMass Dartmouth, and we've been fortunate to have funding from a number of different sources for supporting this work. Thank you very much. I'd be happy to answer any questions if you have them.
I have a question. When you did this atomic force microscopy experiment, so you used a cocktail of the cranberry juice. So you could have used the control as the other component of cranberry juice as a cocktail rather than water. Why did you choose water?
So initially we used cranberry juice or we used water, but the concentration of cranberry juice in the overall solution is pretty small. So and again, we fully acknowledge that's not the ideal control. We didn't have our own placebo, and I don't even have any analytical chemistry equipment. That's not really my area. So we got just enough data using water as a control to be able to go and ask someone for a real placebo beverage.
Thanks.
So the effects of the cranberry juice, you tested them the first time over just a day period with one subject, basically. So that kind of progression would be as if you were taking cranberry juice, a glass of cranberry juice a day.
Right. So in the other study, the 48 hours, it's actually the same volunteer. So that volunteer will drink the juice, give their urine samples over 48 hours. Then we'll have a washout period where they just, eat or drink whatever they normally drink. And then that same person has the other beverage. So if they started with placebo, then they get juice. Or if they started with juice, then they get placebo. So it's a little bit easier to identify the effect when you compare that one person compared to if we just got 20 people to drink juice and then got 20 people to drink placebo, it would be much harder to match the results up. What we learned, this had been my first time doing it, what we learned is that working with urine and working on a clinical study is really, really complicated. We actually I think we started with closer to 40 volunteers, and a lot of them had to drop out of the study for various reasons. Like some people sign up for the study and then they just don't drink the juice, or they come back and they gave you the urine sample but it's the wrong time, or if someone had to go on medication or had some other kind of health issue, they would be discounted from the study. So I don't know if you ever read about clinical studies and you wonder why did they have such a small sample group, or why are the results so hard to interpret. It's just very, very hard to work with humans compared to bacteria. Yes.
I know that you said that this is like an 8 or 9 year endeavor so far for you. And since you've been progressing with it, you've kind of come up with more questions as well as answers. For this next round of trying to find answers to these questions, do you have a target date set for what you're trying to reach when, or are you just kind of playing by eye or ear?
Yeah, I'm sorry, my voice is getting a little weak. I'm going to use the microphone. So what I really think we're getting to the stage where we could develop a new product. And so we have a proposal in right now to the NIH in partnering with a startup company, and working with this chemist that I mentioned. I think we're at the point that if we could identify the right molecules within 2 or 3 years, we should be able to come up with a product. So, it took a really long time, not just me, many other researchers who've been working on this to be able to get to this point, but that's where I'm hoping to go the next time we hopefully will receive money for our work. Yes?
For what purpose? Why did Ocean Spray develop a placebo?
Well, they also do their own studies.
They do their own.
So they have a research lab. They have a really nice research lab and facilities, and they do their own studies that they don't really publish. So we don't know all the results of their studies. But they actually, the NIH asked people to develop a placebo. So maybe around the mid-90s, the NIH, the National Center for Complementary and Alternative Medicine, they got really interested in funding cranberry studies, but they said, we can't really interpret any of these results because all of you are using totally different materials. So the NIH asked people to develop a placebo, and it was Ocean Spray who developed the placebo. And for a while, they were providing that to people all over the country who wanted to do these kinds of studies. Yes?
Now, the packs, are contained in the cranberry tablets that are available? Yes.
Well, that's a totally open question. I think one of the big issues in the industry is that it's not one molecule. It's a whole class of molecules, and they could be polymers of different molecular weight. So there's no commonly accepted analytical tool to say, I have this many packs. And that's one of the things, there's like a cranberry lobbying group. They're trying very hard to get to that point because it's not good for them either. If somebody buys their pills and they don't work, that's not good for them either. So they would like to see a technique developed that everybody in the field would agree upon and say, this is the technique we're going to use, and if we use it, it has to be at least 100 milligrams per liter. So right now, it's kind of fuzzy.
It's the same problem they have with St. John's wort.
Mm-hmm.
Yeah.
Yeah. Yes.
Sorry, so the packs are found in all the cranberry juice, cranberries, and the pills. It's just the amount that the debate is over?
I think so. Yes, I think so. Yes. You had a question?
I was kind of going to ask the same sort of thing. Is like one serving of eating cranberries equivalent to one serving of drinking cranberry juice? But that's, you just answered.
Mm-hmm. Yes, in the back.
You said Professor Meadow from UMass Dartmouth, she's isolated these different compounds in berries. How many combinations are you testing at the same time? Are you looking for a synergistic effect or a single compound?
I think we have about 12 different things planned, and it's really just a question of time and funding. So, what we're hoping to do is a rational approach. So if the first few we test are completely off, then we won't keep going in that same direction. So, you plan this out for a 2 or 3 year period, but then your initial results inform what you do in the next step. Yes?
You mentioned that you have the effect from the cranberry juice, reduces the bacteria adhesions. But then when you put it back into the medium without the cranberry, is the level of the bacteria much higher? Or is it pretty much at the same when it originally was there? I mean, does it come back with a vengeance when there's a problem with MRSA?
I don't think so. I mean, I didn't specifically test if cranberry makes it more pathogenic, but I don't think that's the case. Again, because it's really like a physical thing. It's almost like there's molecules from the cranberry that bind with those proteins and block them from attaching. So,
Okay, thank you.
yes.
I hate to say it, but it would be like if somebody got an injury and then they had a child, the child wouldn't have the injury, correct? Like that's how the cranberries work.
Right, but so there have been some suggestions in the literature that maybe something in cranberry altered the genetic structure inside the bacteria. So if it did, that would be passed down to the daughter generation. At least our evidence has shown that's not what is happening. But yeah, that was a good analogy.
Okay, great. Thank you so much.